Welcome to the University of Pittsburgh's third Virtual Health Academy. Today we will be featuring Dr. Arthur Levine, Executive Director of the University of Pittsburgh Brain Institute. Dr. Levine was the Dean of Pitt's School of Medicine and Senior Vice Chancellor for the Health Sciences for more than 2 decades. Dr. Levine will be discussing cutting-edge research initiatives to discover the cellular and molecular root causes of Alzheimer's disease and other dementia disorders. Before we begin, we encourage all participants to use the Q&A function located at the bottom of the screen to ask questions. You can also use the chat feature to make comments. We will have an opportunity toward the end of the presentation to address as many questions and comments as possible. If we do not get to your question, if you have additional questions, or if you'd like to support our efforts, we will share our contact information at the end of the slide deck and follow up with you personally. Thank you for your interest and participation in today's Health Academy. With that, I will now turn the program over to Dr. Levine. Welcome, Dr. Levine.
Thank you, Anita. And I would like to welcome my audience to the initiative that we have underway in Pittsburgh that we have called an assault on Alzheimer's disease. So let's start with the first slide. Little bit of a technical glitch here. Okay, sorry. Why are we calling this an assault on Alzheimer's disease? It's because we finally need to understand what causes this horrible disease in the first place. Until we knew that a virus caused polio, we couldn't make a polio vaccine. And until we recognized the virus that causes COVID, we couldn't have the miracle of the vaccines that were produced in less than a year. Alzheimer's is a very complex disease, and it has many possible causes, not just one. But until we understand the cellular and the molecular root causes of this disease, I doubt very much that we will be able to treat it curatively, or better yet, to prevent it. So several years ago, when I was still the dean of the medical school, I decided that this is a disease that we should go after. We've been a very successful institution with polio and organ transplantation and so forth. Now it was time for us to take on what may be the biggest challenge of all, which is to try to find a way to understand the basis of Alzheimer's disease and thereby to treat it successfully. And as I said, better yet, to prevent it altogether. Why are we so focused in this way? Someone develops Alzheimer's disease every minute. The economic burden is huge. It's at this point about $300 billion, or at least 10% of all our US healthcare costs. In addition, somebody has to stay home and take care of grandmother or grandfather. That's another $234 billion in cost. So this is not only a horrible disease, and that's what we have to think about first and foremost, the cost to the patient and the cost to the family, but it also is a huge economic cost to the country. The risk of Alzheimer's disease doubles every 5 years after the age of 65, and 1 in 10 people more than 65 will have Alzheimer's disease. 2/3 of those people are women. We have no current or promising agent yet to prevent or to treat Alzheimer's disease. And I'll be saying more about that this afternoon. We have almost 6 million people in the United States that are living with Alzheimer's. And because it is mainly an age-dependent disease, and we know how the population and its aging is increasing arithmetically, we can say that by 2050, there may be 14 million people in this country who have Alzheimer's disease. What is it? There was a painter named William Udermolen who did self-portraits once he knew that he'd been diagnosed with Alzheimer's. And you will see in 1967, this was his self-portrait. And 30 years later, in 1996, when he had the disease had begun, he's no longer able to draw the features in the detail that he had when he was a younger person. Just a year later, he could draw even less. His clothing became simplified, his facial features became simplified. A year after that, he wasn't looking like himself at all. And by 1999, 3 years after his disease was diagnosed, you can see that there was very little left. And 4 years after the disease initiated, he was able to show 2 eyes, but nothing else. He was essentially featureless. And these self-portraits from a fine artist really demonstrate how this disease unfolds. What are the hallmarks of Alzheimer's disease and its progression? How do we recognize this disease clinically? Well, we have to recognize it not only clinically, because there are many forms of dementia, but we have to recognize it specifically and selectively. This disease starts, we believe, with a trigger called amyloid. It's a large, misfolded toxic protein that begins to accumulate in the brain of patients with Alzheimer's disease. I mentioned that it's a trigger. In a way, this is like a barbecue. This is the flame that starts the disease, and then we move on to hot coals. And in this case, those hot coals are called tau. These are tear-shaped proteins that wrap around strands of protein in the cell, but specifically wrap around DNA. I would point out that we all have little bits of amyloid, and in fact, in very small amounts, they may serve a protective function. The same thing is true of tau. We all have tiny bits of tau in our brain, and they may in fact be protective when they wrap around DNA. But when we start making a large amount of tau and changing its chemistry, which is what we think amyloid does to it, then the tau wrapped around the DNA begins to strangle the DNA and break it apart. And when that happens, our cells and the connections between those cells begin to deteriorate. We have an immune system in our brain. We have an immune response. We have cells called astrocytes, which are part of the immune system. And now we have an immune response against these deteriorating cells. And finally, years later, the brain begins to truly degenerate. And so it loses its volume. The white matter in the brain begins to deteriorate. Beta amyloid is present in abundance. And in about a third of patients with Alzheimer's disease, they have expression of a particular lipoprotein called ApoE4. We all have genes for ApoE, and about 10%, maybe 15% of us have one particular form of this lipoprotein called ApoE4 that increases the risk of Alzheimer's disease probably tenfold. So this is the progression, and I'm about to show you a short video which captures what I've just said in 3 dimensions.
Neurons depend for their survival on long chains of molecules arrayed like railroad tracks that transport nourishment across the cell. What keeps the tracks stable are short strips of protein called tau that work like railroad ties. In Alzheimer's disease, the tau mysteriously starts to curl and tangle, causing the strands of molecules to separate and collapse. In the spaces between neurons drift thousands of molecules of protein. In an Alzheimer's brain, some of them are suspicious protein fragments called beta amyloid. Sticky and resilient, bits of beta amyloid clump together to form an expanding mass called a to the outside of the neuron. Sensing a hostile invader, the brain's defensive cells fight back, releasing powerful chemicals. But the counterattack backfires. The chemicals set off a series of reactions that slowly destroy the neuron.
How does this disease progress? It's important here for me to give you a short course, a mini course in the anatomy of the brain. Our brain, looked at sideways, has a frontal lobe, and we use that part of the frontmost part of our brain to carry out executive functions like thinking and planning and problem solving. Some personality and some emotion is resident in the frontal lobe. Next, we have the motor cortex, and that's the part of the brain that lets us move our arms and legs and our fingers and so forth. We have a sensory part of the brain where we experience touch and hearing and smelling and pressure and so forth, temperature. We have a parietal lobe, which is responsible for perception and for making sense of the world, and including arithmetic and spelling. We have a temporal lobe, which is really important in Alzheimer's disease because it's the seat of memory and learning and understanding language. And deep within the temporal lobe, we have a little organ called the hippocampus, which is critical in Alzheimer's disease and is in fact the major seat of learning and memory. In the very back of the brain, we have the occipital lobe, and that's how we see. When we transmit a sight from our eyeball to the optic nerve, it reaches way back to the back of the brain, and that's how we finally form a picture of what it is we're looking at. So with that course in neuroanatomy, we can now move on and talk about what happens and how these parts of the brain are affected in Alzheimer's disease. It starts with mild cognitive impairment, and that's a period that lasts for about 7 years. The disease begins in the temporal lobe, and the symptoms are short-term loss of memory. But I have to caution you here that short-term memory loss is a part of the normal biology of aging. And most people who have short-term memory loss simply have it as they get older. They forget a little bit. They may become wiser. They may have memory for things long in the past, but they don't have Alzheimer's disease. So we have to be able to make that distinction. Some of these people will progress to Alzheimer's, many won't. Over the next 2 years, we have mild Alzheimer's disease. The disease has spread to the lateral temporal lobe, to the parietal lobe. The symptoms now are problems with reading, sometimes problems with balancing the checkbook, poor recognition of objects, maybe getting lost when we drive home from work to our house on a road that we've always known. So that's a 2-year period. Now it accelerates. For another 2 years, we have moderate Alzheimer's disease. The disease now spreads to the frontal lobe, the executive part of our brain. And the symptoms are poor judgment, impulsivity, a short attention span. And finally, we have a 3-year period of severe Alzheimer's disease when the disease ultimately spreads throughout the brain and to the occipital lobe where we have visual problems. So problems with vision and incidentally, problems with smell, with hearing, all of this is part of the disease. So you can see that from the time that we can make a diagnosis of Alzheimer's, it's about 7 years, if we include some part of mild cognitive impairment, 10 to 15 years, unfortunately. We've had one breakthrough in Alzheimer's disease research, but it's unfortunately not a treatment breakthrough. It's a diagnostic breakthrough, and it happened right here in Pittsburgh in our medical school. These two fellows, Bill Clunk and Chet Mathis, spent 20 years trying to invent a chemical, as it were, that could be inserted into the vein in your your arm. And they hoped that if they were successful, that chemical would follow the venous route, end up in the brain and bind selectively and specifically to the, what I call the trigger of the disease, amyloid or beta amyloid. So Bill Klunk is an MD, PhD, member of our Department of Psychiatry, and Chet Mathis is a PhD radiochemist in the Department of Radiology. And after 20 years of working closely together, incidentally, they're great salmon fishing buddies also, and maybe why they were fishing or not fishing, they finally were able to develop a compound which we call Pittsburgh compound B. And that allowed for the first time our ability to see amyloid plaques in the brain in patients who were living with Alzheimer's disease. Until this discovery, Alzheimer's could only be diagnosed, unfortunately, at autopsy, but now we can diagnose it in people who are very much alive. So they infuse this chemical intravenously. It goes to the brain. A person with Alzheimer's disease will have a PET scan that looks like this. This is the brain without the infusion of compound B in the vein. This is the brain with a PET scan to visualize it, and you can see it looks blue. It looks blue in my cartoon, but the brain itself, of course, is not blue. In Alzheimer's disease, however, the brain looks reasonably okay early on, but when we infuse Pittsburgh compound B into a PET scan, you can see all of this yellow and red is beta amyloid. And this is the first time that we've been able to make the diagnosis this way. Chet Mathis and Bill Klunk have won all kinds of prizes and awards for this very important development. Now, when they did that, they were able to look back at people who, for one reason or another, had problems very early in the course of their disease. And when they did PET scans with the infusion of compound B, they showed for the first time that amyloid began to accumulate in the brain 15 to 20 years before the very first symptoms. This was a very critical discovery, an unfortunate one, but a critical one. So over 15 to 20 years before the very first symptoms of memory and learning and cognition, amyloid was already accumulating. And at the same time, it was triggering an accumulation of tau, and in a funny form of tau. Tau, remember, is wrapped around the DNA. It begins to form tangles. It tangles up the DNA, as you see in the blue line. So this was happening in this 15 to 20 year period. And most alarmingly, while this was happening, the connections between our brain cells, strands, very thin strands, like tiny little pieces of hair that connect one neuron to the other, they were beginning to deteriorate, to break apart. And that meant that we were losing the connections between brain cells. So I mentioned that the symptomatic period might be 7 to 10 years, maybe a little longer in some people. So this whole period of time follows something that's happening in the brain 15 to 20 years before. So the disease, as I mentioned, is largely age dependent. So it probably starts perhaps around the age of 60, and symptoms may develop around the age of 75 or even 80. Now, in one of my early slides, I also said that one of the hallmarks of this disease was a lipoprotein called ApoE4, a cholesterol transport protein. About 1 in 10 people are born with the gene that encodes this lipid-carrying protein. And If you have a double set of those genes, one from your mother and one from your father, the likelihood that you're going to develop Alzheimer's disease is very high. And in fact, more than a third of all patients with Alzheimer's disease, unfortunately, were born with one or two copies of the ApoE4 gene. It doesn't help us very much to know that because we can't prevent the disease. But someday we will be able to prevent it, and then we'll be able to address people who unfortunately have been born with that particular gene. It doesn't mean that everybody who has it is going to develop Alzheimer's disease. Maybe 1 in 10 people will that have the gene will develop it. If they have both genes, it'll be faster. They may develop it younger, but that's the only gene that we know with certainty is responsible, at least in some patients, for the development of Alzheimer's disease. I've already mentioned that age is a driver of Alzheimer's disease. I think you know that very quickly, just to show you that around 65 or 70, maybe a little later than that, we begin to see the symptoms of Alzheimer's disease, but 50 to 20 years before that, we begin to see amyloid developing in the blood, in the brain. We see tau changing its chemistry, and we see cognitive impairment. So it's all age dependent. One possible cause of Alzheimer's disease is damage to the DNA in our brain cells. That happens to be a particular interest of mine. I have a laboratory myself in the Brain Institute, and we work on the possibility that damage to DNA could be driving this disease. There may be many causes of Alzheimer's. This could be one of them. But DNA damage comes about normally. Every time we eat food and have metabolism of that food, we produce radicals of oxygen, and oxygen radicals damage our DNA, just like cigarette smoke can damage our DNA or sunlight can damage our DNA. But as we age, we have a lifetime of oxidative metabolism, like rust. It damages our DNA. And unfortunately, as we get older, we also have less ability to repair that damage. We have exquisite mechanisms for repairing DNA damage. Many genes and proteins are involved in recognizing DNA lesions and fixing them. But as we get older, that system of repair begins to deteriorate. So if that happens in the brain, one possibility is that is at least one possible cause of the disease. But we've identified a lot of genes that may have contribute to some of the Alzheimer's risks. We've talked about genes that produce the trigger amyloid. PSEN1 is one we'll come back to. We talked about ApoE4, the protein that carries cholesterol, and how that accentuates the risk of Alzheimer's disease. Then we have a whole bunch of genes that contribute to the formation of tau, that contribute to the immune response of those astrocytes and their chemicals and so forth. So you can see just how complex the total risk portrait of Alzheimer's might be. We don't yet know which of these genes are critical, with the exception of the genes that produce an excess of amyloid and the genes that are responsible for those people who have the that were born with the ApoE4 gene. The rest of it is all speculative at this point, things that we can identify, but we don't know with certainty how they contribute. Another little anatomy lesson. This stick represents what the precursor of amyloid beta might look like. It doesn't really look like a stick, but in the cartoon, it helps us to understand it. We have a good enzyme called alpha secretase in the membrane that wraps our cells. And if the stick encounters alpha secretase, it breaks down into perfectly harmless parts of the stick. But if in fact it goes in the opposite direction and encounters an enzyme called beta secretase or base 1, it begins to go in a bad direction. The stick begins to break down. It then encounters a really bad enzyme called gamma secretase. Both of these enzymes are in the membrane that wraps around the cell. Alpha secretase is inside the cell. So it depends which direction the stick, again, is just a cartoon metaphor, which direction it goes in. But if it goes from beta secretase to gamma secretase, eventually this large precursor stick breaks down into molecules of amyloid. Amyloid molecules are inside the cell. They can aggregate around the outside of the cell and form a plaque. And now, hopefully, you have a sense of how this whole process unfolds. Well, many scientists, pharmaceutical and biotech companies know about, have seen this cartoon. They know about how these proteins unfold. And so they've tried to inhibit various aspects of this, what might be called an amyloid generating process. But there are other parts of it. The energy factory of the cell has been speculated as a problem. If it's damaged, we talked about the bad enzyme, gamma secretase. We've talked about immunity and inflammation. That might be a target, and amyloid beta itself. Clinical trials have been done in many parts of the world over many years. Every one of them has had a negative result. We've never found anything in any biotech or pharmaceutical company or anywhere else that in fact altered significantly the course of Alzheimer's disease once it's underway. Most recently, Biogen produced a protein, you've probably read all about it, called aducanumab. It's a monoclonal antibody that attacks beta amyloid. And in fact, it does a good job. It actually begins to remove beta amyloid from the brain. It made no substantial difference whatsoever in the clinical course of this disease. I don't believe that it's a biologic failure. I believe that it's a timing failure. If in fact we had been treated, if people had been treated with this antibody 20 years before the first symptoms, we might well have prevented the disease. But we can't do that with the whole population. We might be able to do it with people who have the very bad ApoE4 genotype, but we don't even know that. In any case, you probably know that the drug has, despite its marketing, has essentially been a failure like all the other proposed treatments for this disease. So where do we go from here? Well, scientists are really like the blind men trying to say what an elephant looks like. So here's a blind guy that only feels the ear of the elephant and thinks that the entire elephant looks like an ear. Here's a guy that feels only the tusk and he thinks the entire elephant looks like a tusk, and so forth. So that's the situation we're in. This is a very complex disease. Many genes, if mutated, may contribute, or just damaged, may contribute to the disease. We only know with certainty about ApoE4, and even that is uncertain. You can have ApoE4 and still not get Alzheimer's disease. Remember that I said it might be 1 in 10 people with that particular gene. But we know about another gene that produces beta amyloid when it's mutated, and that's the PSEN1 gene that I showed you in the earlier cartoon. Knowing all this, what do we do next? Well, all those clinical trials have failed. There is no drug that significantly alters the course of the disease and certainly doesn't prevent it. So we need to, because we can't start doing clinical trials in people who have no hint of the disease, we can't wait to do studies in human beings, because if the disease begins 15 to 20 years before symptoms, that's a huge problem. Secondly, it costs a lot of money to even screen a patient with memory loss or a little bit of cognitive impairment to enroll them in a clinical trial. We have to do compound B and a PET scan. They have to be willing and able to enter clinical trials. So it's very difficult to mount a robust clinical trial in Alzheimer's disease, and also very expensive, as you can see. Plus which, I mentioned that there may be many risk factors. I mentioned my own interest in DNA damage is one. We talked about ApoE, and we're going to talk about PSEN1, and there are probably many other genetic variants. Remember the slide that I showed you with those many genes that have been implicated one way or the other. Viruses. I do want to stop for 1 second and say a little word about COVID, because you're probably all familiar with what we're calling long haul COVID. People who have an acute infection and end up 6 months later with what we're calling brain fog, memory problems, learning problems, and so forth. And we have been able to see in the brains of patients who have died of COVID, at least a number of them, perhaps 30%, that the COVID virus is in their brain, as is the immune response. So I worry that this, in fact, could be a prelude to a lot of people who had long haul COVID infection eventuating years later in Alzheimer's disease. And there may be other viruses as well. I talked about DNA damage, hypertension. People who have out of control hypertension for long periods of time probably have some increased risk of Alzheimer's. We talked about ApoE. Sleep deprivation is interesting. If you take a mouse and don't allow the mouse to sleep for 48 hours, it develops a lot of amyloid in its brain. Head trauma. I worry about children and teenagers playing football. They may wear a helmet, but the brain still shakes around inside the helmet. And I worry that some of these people may in fact have a higher risk years later of Alzheimer's disease. Systemic inflammation. Some of the bacteria in our gut may secrete products that go to nerves that eventually go from the gut to the brain. So all of these are possible risk factors, and we have to explore all of them. And that's what our assault on Alzheimer's implies. Now, I just mentioned the mouse and the fact that if you keep it from sleeping, it will develop amyloid in its brain. But mice don't get Alzheimer's disease. So they also don't play the fiddle. But the literature is filled with thousands of papers that talk about trying to replicate Alzheimer's in mice and rats. And yet, every time we've tried to use a drug in a mouse or a rat, or even to predict the development of Alzheimer's, we failed. Why is that? Well, there's a simple explanation. Here's the human brain in a side section showing parts of the brain that are involved in Alzheimer's disease. Remember the frontal lobe, the temporal lobe, the hippocampus. Let's go here to the other end of the slide and look at the rat or the mouse. And you can see that it's completely missing those same areas of the brain that we know are afflicted in Alzheimer's disease. So it's not going to be an adequate model for really trying to find a drug, for example, that would prevent the disease. It just doesn't have, through evolution, the same parts of the brain that are critical, because they're the parts that are targeted in Alzheimer's disease. However, non-human primates do have the same parts of the brain. As I'm sure you all know, non-human primates are very similar to human beings. They're within 1% genetically of looking like humans. And in fact, there's a particular non-human primate called the marmoset. It's a very tiny monkey, about the size of a squirrel, that has the very same parts of the brain that the human has that's afflicted with Alzheimer's disease. So we're now going to concentrate on this as an animal model. Well, I've already said that rodents are not an effective model for studying Alzheimer's disease, just because their brains are so different that they really do not develop or mimic Alzheimer's disease. And I mentioned the marmoset. So it's small size, they're social creatures, they have a sophisticated form of communication, they have a way to cooperate socially. The females deliver twins twice yearly, so it's easy for us to breed them. And in fact, if they're identical twins, one might be an experimental model and one a controlled model. They sexually mature by 1.5 years. They usually live about 9 or 10 years, but some of them can live as long as 12 years. And in fact, a few marmosets that we've seen in our own marmoset colony of this age have developed what looks almost identical to Alzheimer's disease in people. That even might begin, if we really look hard, which we haven't done until recently, at 8 or 9 years. Finally, we know, and in a moment I'll show you why we know it, that a particular mutation in a human gene called PSEN1 reproduces the extravagant accumulation of amyloid. When that happens in people, they can develop Alzheimer's at a very early age. And in a moment, I'm going to tell you about early onset Alzheimer's occurring in families. But for the moment, let me just tell you that our ability to edit genes, and you've probably all read about CRISPR, the technology for which the Nobel Prize was awarded a few years ago. When we use that to edit the genomes of marmoset embryos, we can reproduce Alzheimer's disease in the marmoset. And because we know that if marmosets live long enough, they may naturally develop Alzheimer's, we probably have a solid rationale for this particular animal model. So why does a person develop Alzheimer's disease? Well, the sporadic form, the age-dependent form, is most of it. But there are families, fewer than 5% of Alzheimer's patients, where everybody in the family will eventually develop Alzheimer's disease. And it's because they have that PSEN1 mutation. It's a mutation in that last bad enzyme, gamma secretase, that in fact leads to the hypersecretion and the extravagant accumulation of amyloid in the brain. So as I said, this PSEN1 mutation leads to early onset Alzheimer's in human families. We can replicate it with genetic engineering in the marmoset. And we believe that the time course and the signs and the pathology will mirror the onset and progression of the age-dependent form of Alzheimer's disease. Very quickly, this is what a curve would show in the familial early onset form of Alzheimer's, the rare form, just a few percent. Again, they have the 15 to 20-year period before the symptoms. That begins in their 20s. They usually die in their late 30s or early 40s. The common age-dependent form looks identical. It starts later. Maybe amyloid begins to accumulate in the 60s. They may die by their 80s. But the point of all of this is that by studying families with the rare early onset form and marmosets that have been engineered to replicate that form, we will learn a great deal about the common age-dependent form. So our approach in Pittsburgh is to study all these complexities in one place. We now have young investigators that we've recruited just in the past few years from Harvard and MIT and from the Max Planck Institute in Europe and from the NIH, a wonderful group. We have 9 such new investigators. We're searching now for a 10th, having composed one of the strongest groups in the world to study the basics of Alzheimer's disease. Some of them are studying amyloid and tau. Some of them are studying how we get rid of these bad proteins, if we're able to, in the brain. Some that are studying As I said, the immune response, if we could get rid of that, it would help. We have others studying ApoE and its role in the cholesterol pathway and how that intersects with Alzheimer's. I mentioned my own interest in DNA damage. I mentioned viruses like COVID. I mentioned the fact that part of Alzheimer's is a compromise of vision or smell or hearing, other genetic environmental factors. So we have now 9 young investigators and others who study Alzheimer's part-time. But here we have 9 new young full-time Alzheimer's investigators, about to have 10, trying to put all this together. It's such a complex disease. So many genes may be involved, so many environmental factors as well as genetic factors, that we need this kind of workforce. This is sort of a Manhattan Project. For those of you of the generation that remember what the Manhattan Project was and the development of the atomic bomb, we're trying to apply that same kind of intensity and group intelligence to this horrible disease. In general, I would point out that we have a lot of neuroscientists at the University of Pittsburgh, more than 150. And although they may not all work on Alzheimer's full time, they work on the brain. We have a brain bank with now almost 1,000 donated brains. We have clinical and imaging data for more than 5,000 patients. We have world-class expertise in engineering genetically and engineering tissues of the brain. We have a center of excellence for applying artificial intelligence and machine learning, theoretical computation to all that we're doing. We are one of the country's leading centers for studying the brain in non-human primates. We now have, incidentally, about 220 marmosets in the very building from which I'm speaking. That's probably 10% of all the marmosets in the country. And we have a large community for doing brain imaging and recording. So let me finish by pointing out that we are the very institution, and you know this well, where Jonas Salk stopped polio. You may know not quite as well that Peter Safar, who founded our Department of Anesthesiology and Critical Care Medicine, was the man who first developed CPR. You may also not know that Bertie Fisher, one of our surgeons, who grew up in Squirrel Hill, went to Taylor Alderdice High School, went to college here, went to medical school here, did his residency here, was the first person to recognize that breast cancer is not simply a localized disease. And in all too many women, it already has begun to metastasize in other organs of the body at the time of diagnosis. And therefore, doing a radical mastectomy, a barbaric procedure, is inappropriate. But giving chemotherapy after treating the lump is the way to approach breast cancer. And that resulted in the saving of hundreds of thousands of lives and is a lesson for all of cancer. Another one of our stars. And then finally, and you all know Tom Startzel, who turned first liver transplantation and then all of transplantation into a reproducible clinical entity. So we've done it before, and if you wish us luck and perhaps help us support this research, I feel quite confident that ultimately we will do it again. Thank you.
Thank you so much, Dr. Levine, for your thoughtful and thorough presentation. We've received quite a few comments and questions, so we've taken the liberty of choosing a few that follow some common themes. First of all, is there any potential for predicting who will develop Alzheimer's disease? And is there any way currently known to modify that risk?
A very good question, Anita. As I mentioned, there are a small number of human beings who are born with the gene for the cholesterol-carrying protein ApoE4. Now, there's no guarantee, even if they have both of those genes, one from mom and one from pop, there's no guarantee they're going to develop Alzheimer's, but they do have a high risk of doing that. We also know in the families where everybody develops Alzheimer's that we can sequence their genome, we can find the mutation for PSEN1, and we know for sure if they have that mutation, they're going to develop Alzheimer's disease. So with that knowledge, once we do further research in the marmoset and hopefully find a way using that animal model to prevent the disease altogether, we would finally have a drug or even a vaccine that would prevent it.
Thank you. Another question we have is there a biologic reason for why women are more likely to develop Alzheimer's disease than men?
That's an excellent question. I don't know the answer to that. A simple answer would be women live longer than men. And because it's an age-dependent disease, maybe that's why they have more. But biology is very complicated, and simple explanations are nice, but they don't always give us the right answer. So there may be other biological reasons that have to do with the production of estrogen or the waning of the production of estrogen that could be involved. There may be reasons that have to do with the cardiovascular system in women as opposed to men, or the immune system. Women in general tend to be more responsive immunologically than men. That's why there's more lupus, for example, in women. So I can think of a lot of reasons, but all I can say is that they remain speculative.
Other questions? Dr. Lopez takes care of my mother at the Alzheimer's clinic. Do you and your colleagues work with him and the clinicians?
All of our colleagues work closely together, but Dr. Lopez will be the first to recognize that until we understand what causes the disease in the first place, when we can really identify targets for curative or preventive treatment, we have to work in the laboratory while Dr. Lopez and his colleagues work with patients in clinical trials.
Thank you. A couple of other questions that have come in. Do you think alcohol consumption plays any role in Alzheimer's disease and its effects on the brain?
Well, we know that alcohol affects the brain. I don't know that alcohol plays a significant role in the development of Alzheimer's disease. It's one of the many variables where, together with other variables like one's genes or one's diet or how much sleep one gets, all of these things may in some way contribute. But once again, it's too complex a disease because the brain is so complex. It's too complex for us to immediately have a calculus for alcohol or addictive substances or anything else. All we can do is try to do research. Now, the marmoset would be a great model. In fact, we could expose the marmoset infant to small amounts of alcohol and see whether they have a higher incidence ultimately 2 or 3 years later of Alzheimer's disease. It's an important point, incidentally, that I wanted to make, which is that the virtue of the marmoset model is that if they develop Alzheimer's, they're going to do it very quickly. It's not going to take them 15 to 20 years. We'll see it in 1.5 years or 2 years. We're already seeing amyloid in their brains in infancy.
To that end, why has it taken so long to find a cure for Alzheimer's disease?
Well, I would like to say we haven't been lucky, and luck does play some role in science, but mainly because of the complexity of the brain. It's by far our most complex organ. We have millions and millions of brain cells. We have millions of immune response cells in the brain. If you think about the little hair-like strands called synapses that connect one brain cell to another, we have a huge network that's far more complex than even the most complex computer. And if we begin disrupting those strands, those synapses, then we lose the ability to even understand how this network is operating. So what I would say is that we're dealing with a far more complex intellectual challenge when we work on Alzheimer's disease, even than when we have tried to address cancer successfully. It's just very complex, which once again is why I made the decision when I was the dean. And while hopefully I'm a role model, having given up cancer research for brain research myself, to take on this huge intellectual challenge.
Thank you. We also got a question. I'm not entirely sure in which sense the questioner meant it. Do you think that marijuana has an effect or contributes to Alzheimer's? I'm going to add to that, is there a role for medical marijuana in Alzheimer's disease?
Well, let me take the second part first. There may be a role for medical marijuana. We have some evidence that medical marijuana has been useful for one disease of the brain, and that's infantile epilepsy, Dravet syndrome. That is in fact, the only clinical entity where we have certainty that medical marijuana is useful. But we ourselves are engaged in clinical trials of medical marijuana for other diseases, not yet Alzheimer's disease, but other clinical trials for other diseases and disorders to see whether medical marijuana is safe and effective. We don't know that yet. Nobody knows that yet. Now, back to the first part of the question, could it have anything to do with Alzheimer's disease? All I can say about that is that anything we put in the brain or change the brain or influence its chemistry or biology could potentially be one of those possible risk factors, but we don't know that with any certainty. I do feel strongly that it's a mistake for so many people in our population, particularly young people, to be on marijuana, medical or non-medical, for years of their lives, because I know for sure whenever we put anything in the brain that affects how the brain works, there is some danger ultimately, maybe decades later, of having harmed the overall function of the brain.
Thank you. One other question, well, actually two. One is, how do we support this effort to cure Alzheimer's, prevent Alzheimer's? What are your next steps in achieving that end?
Sure. The immediate next step, and by far the most important one, is for us to, in fact, make our understanding of Alzheimer's disease known to our friends and our neighbors and our relatives, the people we work with, our employers. There's a lot of mythology about Alzheimer's disease, and we've already seen from what's happened in this country with regard to how we talk about masks and vaccines that the influence of science and science education is critical to our understanding how we deal with our own health and the health of other people about whom we care. The same thing is true in Alzheimer's disease. If we want to do something about this disease, we have to make a lot of people aware of the kinds of things that I've tried to talk to you about today. That's even more important than money. But of course, I would have been a bad dean and a bad director of the Brain Institute if I didn't say something about money. Of course, research is expensive. Every time I bring a new young investigator from Harvard or MIT or the Salk Institute or Hopkins or Berkeley to Pittsburgh to work on Alzheimer's disease, it costs between $1 and $3 million. Not just for the salary of the young investigator, but to renovate a lab, to buy lab equipment, to let them have technical help, to let them have graduate students and fellows, and to do that for several years before they have good enough results to actually leverage their results and get a grant from the NIH or the National Science Foundation and so forth.
And on that note, we're going to bring this discussion to a close. Thank you so much, Dr. Levine, for leading this Health Academy. Thank you so much to our audience for taking the time to learn about the work we've been doing here at Pitt. You will see on the screen contact information for the Brain Institute at the University of Pittsburgh. Please feel free to reach out if you would like to learn more, or if you would like to learn about ways to support our research endeavors.
Just one correction, Anita.
Yes.
I didn't want to leave our wonderful audience thinking that they had to give us between $1 and $3 million. That I want to point out that the Cathedral of Learning was built with dimes, and Dr. Enders at Harvard accepted pennies when he was trying to develop a cell culture for poliovirus. So anything would be wonderful. Thank you.
Thank you, Dr. Levine, for the clarification. And again, thank you all for the questions that you've inserted in the queue. We will respond, even though we could not do so live. Again, reach out. We'd love to hear from you, and have a wonderful day.